Okay, everyone, welcome. I'll just give a brief overview while we wait for a few more people to trickle in. My name is Anna, and I'm a scientific account manager here at BSI. We're excited today to dive into a discussion on peptide sequencing using traditional and novel AI-based algorithms. Our presentation today will be led by Christina Yurchik, our product care manager. Proteomics research is evolving rapidly, and with each new version of Peaks, our goal is to provide you with the tools you need to stay at the forefront of discovery. Accuracy and sensitivity are key, and Peaks offers unique solutions with state-of-the-art sequencing algorithms seamlessly integrated with database matching. This approach not only enhances the confidence of identifications, but also allows for deeper profiling of post-translational modifications and mutations. Furthermore, by leveraging deep learning to predict missing fragmentations, Peaks excels in identifying novel peptides and proteins from incomplete databases, making it the preferred method for modern proteomics research. Today, Christina will present for 45 minutes on our sequencing technologies incorporated in Peak Studio 12.5, and then we'll follow with an interactive Q&A session to address any questions you guys might have. And with that brief introduction, I will hand it off to Christina.
We'll be focusing more so on the peptide sequencing in Peaks and some new developments, and then how we take advantage of these algorithms to enhance confidence and identifications in canonical and the non-canonical peptide matches. Before we dive right into the sequencing, I would like to take a step back just to remind you that at Bioinformatics Solutions, we have a series of products focused on delivering versatile solutions for various applications in proteomics. And in terms of the software, we have a one-stop kind of complete bottom-up solution for proteomics through our Peak Studio and online, which are our desktop, and server-based platforms. We have our PeaksAB intended for antibody and non-antibody sequencing, and then also in-depth really characterizations of PTM, glycan, and mutations profiles. And lastly, we know that the glycoproteomics is truly a particularly complex field of proteomics, and we have our Peaks Glycan Finder that offers a comprehensive solution for characterizations and quantifications of glycopeptides. And for those who may not have mass specs at your institutes, we do offer versatile lab services. If you are in need, feel free to reach us out as well. Now, last time, I don't know if you joined for Natalie's talk, where she gave a very nice overview of our latest Peaks 12.5. And I'd just like to quickly kind of re-highlight some of these key features for, again, our studio and I guess online. Really, all of our software is vendor neutral, so we support raw data loading from different vendors. Studio, again, is discovery and targeted proteomics support, a one software package for DDA acquisitions, for DIA acquisitions, and as of lately, PRM. We have a very nice hybrid DIA with PRM methodology from Thermo that we're supporting as well. And then we're workflow really driven analysis for a variety of applications. What we offer in terms of DDA, again, Natalie talked about it last time, but today I'd like to focus more, at least to start off, on peptide sequencing algorithms. And in our latest version, you probably noticed that we do have two options now for sequencing of peptides. The two options really are two very different algorithms. So the first one is DeNovo sequencing. This is our traditional sequencing algorithm that we developed back when we were starting over 20 years ago. And in fact, de novo sequencing is something that Peaks is well known for. Now, a couple of years ago, we started implementing another algorithm called DeepNovo, and the name itself may give you a hint. It is a deep learning based sequencing algorithm. Now, for some of you, the DeepNovo may be grayed out. And the reason for that is that deep learning algorithms are highly computationally demanding. And in fact, we only allow these to run if a compatible graphic card or GPU is available. And this can be verified through the configurations tab, performance configuration, and if a compatible GPU exists, you will be able to click and select it. And I'm just listing some of these key requirements for compatible GPU. If you have any questions or any issues, just let us know and we'll help you with this. In terms of our traditional de novo sequencing algorithm, the details were first published in 2003, and this algorithm has been pretty much kept as is and used in Peaks, and we're very happy with the accuracy and the performance, and it is really our golden standard to compare all of our new developments to. Essentially, the way it works off of the dynamic programming to find the best path. So what this means is that in some sort of simplified terminology, we look for first in a typical tryptic digest or a specific digest, you look for the Y1 ion. For example, in this case, R or K, which are characteristic of that enzyme or enzyme specificity, and then essentially the algorithm moves on to find the next best match along the path of building up a peptide sequence. And essentially, it is something that you would do manually, but with some here weighting of each next ion match. And then, of course, we take into consideration the B series ions, neutral losses, ammonium ions. Now, for the deep learning algorithm uses prediction models to predict the sequence and then match it to the MS2 spectrum given the fragmentations and intensities. So instead of finding the best path, here the developers talk about a translation problem in terms of how well our model can predict each fragment or fragments that would occur in MS2 for the given peptide sequence. So in 2023, we published the details on our latest deep learning algorithm called GraphNovo, which is now fully incorporated into Peaks. And one of the key advantages of GraphNovo is the predictions of the missing fragmentations and then mitigating these while confirming a peptide sequence. So we know that certain amino acids, particularly hydrophobic ones, will be less likely to produce fragments at the peptide bond. And this also depends on the before and after amino acids. And GraphNovo kind of takes all of this information to predict the best possibility and then correlate with the MS2 information. And just the last thing I want to emphasize is that the final scoring for the peptide sequence will also look at the correlation of then predicted retention time and empirical retention time. So in addition to the fragments with intensities, we also correlate the predicted retention times. And then to conclude this part, basically the DeepNovo is expected to result in a higher accuracy sequencing. However, please be cautious because the results will depend on the sample to model kind of agreement. To note, our models were trained on large data sets. They are vendor specific, meaning we have separate models for the Bruker, so trained models for the Bruker data, for Thermo data, for SciEx data, but still there may be always some overfitting, some caution to be taken with the AI-based algorithms in general. In terms of scoring and how Peaks reports the scoring results, regardless if DeNovo or DeepNovo, we will report a confidence score that you can find under the peptide or DeNovo in this case tab. And essentially, we first calculate the LC, so local confidence for each of the amino acids, based, of course, off of how well we could find each of the ion pairs to define that amino acid. And then we simply take the average across these local confidences and report it as the percent ALC or percent average local confidence. Now, while we do report the percent ALC for both DeNovo and DeepNovo sequencing results, these values do have different weights. And to give you an idea, I'm just showing you here a graph from the application note that we published. You can find this through our website in the DeepNovo section. But essentially, we compared our traditional, in this case, we just call it peaks, but it is the de novo sequencing, traditional sequencing algorithm to the DeepNovo deep learning based algorithm based off of the amino acid accuracies. So these amino acid accuracies are determined through a confident database search matching first. But essentially, if you look at these distributions, you can see they're different. And for example, if you impose a 95% accuracy for the peptide sequence, you can see that the score cutoff here for the deep learning based algorithm is around 55. Do note that this is for timsTOF data specifically. We do have different graphs, somewhat different distribution for different vendors, but the same accuracy for the de novo sequencing would be at around 80 of the score cutoff. So just generally speaking, we recommend about 80 to 90% score cutoff for de novo sequencing, and then about 60 to 70% score cutoff for DeepNovo sequencing for kind of the same high accuracy of the peptide sequence. Alrighty, so now when it comes to the proteomics and LC-MS/MS analysis, the aim of most studies is to really identify the peptide and then the protein given a certain database. And what is exclusive to Peaks is that we take advantage of the sequencing algorithms and then incorporate these for an increased confidence in database search matching. So let's first take a look at a classical general proteomics DDA experiment. You know that we always do de novo sequencing as the first step. And this result is then used for some tag searching for protein shortlisting, which then allows us for an improved search space during the peptide spectrum matching. And then in the identification results, what we will report basically is the peptide score, PSM score for, again, each identified peptide. And there's many different parameters that are involved in the calculation of this score, but I just want to point out that one of the parameters is, in fact, the similarity to the de novo sequence derived by this algorithm. Now, if you look at the actual Peaks output, in the peptide tab, you will see that score as the minus 10 log P of the peptide for each, again, PSM. And again, the de novo sequencing is already implemented into this score to indicate the quality and really the confidence of the peptide spectrum match. I'm also showing you here some of these distribution graphs, which you can find in the summary result, which again will be geared to more confidence given the de novo assisted or assistance from the algorithm. Now, of course, with de novo included in the result, the processing times will be longer, and this has been a debate many times at our company, but we still insist, I guess, on delivering increased confidence over these increased analysis times. And in addition to enhancing that peptide match confidence or the score, we also now have these partial sequencing tags, de novo tags, which essentially we can take advantage of and do some in-depth PTM and mutations profiling. So the way we do it is take the good scoring de novo tags, right? So let's say that one part of the sequence has been sequenced at a high kind of confidence, but the other maybe N-terminus is not really matched to anything given the database, initial database search parameters. So we know that the database search has a limitation in terms of the search space and for example, the number of PTMs allowed before that search space, of course, explodes. However, having these de novo tags, we can now permutate in pretty much all of the built-in modifications in order to fit them in and match this missing, perhaps, part of the sequence. And I have an example here where I pulled a scan with a de novo sequence peptide, which was not found or identified in the database given those initial search parameters, whatever they were. So we have the good sequencing score for each of these amino acids on the C-terminus. However, I even indicated just the N-terminus with the mass tag because really, again, with these search settings or the modifications provided initially, we couldn't really match this part of the sequence to anything. Now, what we have, of course, post-database search, you can select the PIX-PTM algorithm, which will now or can easily really search through all of the PTMs as per Unimod database and as built into our peaks, or you could give it a selected list of modifications, but you can add as many as you wish, essentially, because we, again, based on the sequence tags, now we can, again, permutate in and try to find this missing, essentially, part. And now if you look at the same scan in the PIX-PTM result, again, the C-terminus has been confirmed now in the actual database, right? But also the full sequence has been confirmed with now carbamylation as a modification on the very N-terminus. And also with an A-score of 1,000, which means a very, very high score, very high confidence, of course, in this site of this modification. You can't really see here, but essentially what happens if we zoom in, we see that now the B islands are well matched, again, given this new information that we have or opened up search space, and we can nicely now, at a high score, identify this missing modified peptide. In a similar fashion, spider works. So our spider algorithm can also be checked off, and it will actually look at the Novo sequencing tag as well as the database peptide, and it will be looking for a couple of things. One is the sequencing error, which essentially happens when you have those missing fragmentations. So you may be swapping the two amino acids, or you don't really know, maybe the mass tag is of the same mass as other 2 or 3 amino acids. So basically, we correct for the sequencing errors based on the database peptide, but we also then look for any of the amino acid substitutions or mutations. And to conclude this part, the Novo sequencing is incorporated in our PEAKS database searches, on one end for an improved peptide identification score, and also for additional profiling of modifications, as well as possible amino acid substitutions or mutations. And just to clarify, when we report the results, each additional step is accumulative, meaning that PEAKS PTM result will contain all of those database search matches and the additionally found modified peptides. If SPIDER is added post both of these steps, then all of these results will be included in the newly found mutated peptides. All right, so this is our approach to general proteomics searches, where we take advantage of the DeNovo sequencing. So let's now talk a little bit more about our DeepNovo, our novel deep learning-based sequencing algorithm, and its incorporation for improved immunopeptidome analysis. Again, especially our latest deep learning algorithm called GraphNovo is highly computationally demanding, and it does require a GPU. And then in addition to those mentioned vendor-specific models, we also have triptych and non-triptych models available. So non-triptych models are heavily trained on HLA type 1 and 2 data. So if in PEAKS you select a DeepNovo as a standalone, so this is standalone sequencing algorithm, one thing you may notice in data refine is that we exclude some of these pre-processing steps, because this just better aligns with our deep learning models compared to the DeNovo. And then in the main parameters selection window, you can select either none for now or trypsin, and some modifications. And then you move on to the report filter. And here you will see something, I guess, new or something different. So in addition to our classical percent score filter for sequencing of peptides, we now also have an FDR control. So a false discovery rate for peptide sequencing. And I guess the question is, how is this possible to get the target decoys, and also really why do we need this? Well, to evaluate just in general terms peptide sequencing, we use certain metrics or certain controls, and the typical are peptide accuracy, amino acid accuracy, and fragment ion accuracy. Now, in order to measure these, you need what we call a ground truth peptides, which are really the database search pre-confirmed peptides. So you can imagine that this approach suffers from a bias towards the high quality MS2 spectra, because only these will be contributing to the evaluation of your sequencing algorithm. And this is where our developers came up with an unbiased data-independent sequencing control through the FDR control. And we have a publication last year that we published. So if you want to take a look, please do so. But of course, the question is, how do we obtain a target and decoy distribution, knowing that in the database search approach, you have your decoy peptides, which are simply generated from or by randomly shuffling or reversing the target protein database. Now here, in our approach, we don't have a database, so rather than creating the decoy decoy peptides, we create the decoy MS2 spectra from the target MS2 spectra. And in order to kind of successfully do that, there are several conditions that we need to satisfy, but the two probably most important are while we are adding those random masses, the decoy and target spectra must have the same number of peaks. And the other thing is we need to ensure very similar M over Z and intensity distributions of target peaks and decoy peaks. And with this in place, we now believe that our sequencing tool, if it's a good tool, should really predict a meaningful sequence or peptide sequence with a high confidence score from a target spectrum, but only an arbitrary sequence with a low confidence score from a decoy spectrum. And in fact, this is a graph from our publication. We are able to create these decoy and target distributions, maybe not as beautiful as in my previous slide. However, if you take a look at this graph here, which is also given in the publication, where we basically plotted distributions of the target, decoys, correct peptides, and false identifications, you can see the target and those correct peptides, those distributions align really nicely. And similarly, the decoys with the false identifications also align together. In peaks, what this looks like in our output result, here's just a very simple BSA tryptic digest with kind of the strict FDR 0.1%. Basically, you can see that the FDR curve is completely leveled off to whatever number of peptides sequenced, and then it starts leveling off at 0.1% and kind of slowly goes really up after 1%, which is the expected behavior. We'll also plot the target decoy distributions, and if you take, say, a high kind of confidence score, you can see that the peptide is really nicely sequenced. In this case, target score, right? It's a nicely sequenced YB series of ions, prediction correlation between the empirical and predicted spectra are also in good agreement. The one thing I would like to note is that if you are doing a standalone peptide sequencing and wish to use the FDR control, the analysis will take longer time due to the time needed to create all of these decoy MS2 spectra. So definitely expect some increased analysis times. And just one more thing, which is another figure from this paper. Now that we, of course, have this unbiased control for peptide sequencing, we wanted to compare the performances of our DeNovo, our initial DeepNovo sequencing algorithm, our current GraphNovo, and CasaNovo is one of the other vendors, very nice deep learning based algorithm as well. And of course, we did it across very different sample types. And this is the key whenever you're working with the deep learning, to have the models that can handle very different sample types. And in this case, if you look at our GraphNovo for human, it actually really nicely, it's quite flat for quite some time with the number of PSMs being true, and it's similarly in performance to CasaNovo. But you see when the deep learning may not agree too well with a certain sample, in this case, CasaNovo actually is not doing so well, particularly for a planned sample. For HLA, CasaNovo, again, really good. GraphNovo, really good. And actually, GraphNovo, again, for non-tryptic general proteomics, is performing quite well. So I hope that with this little rant on the sequencing and this unbiased, especially way of assessing the peptide sequencing, I could convince you that we have a very good high accuracy deep learning based sequencing algorithm. So the next question is, how can we take advantage of this DeepNovo sequencing? Now we know that traditional database searches use some sort of canonical databases curated with confirmed known proteins of known functionalities and widely expressed. However, this limits us in the discovery of and validation of anything novel and novel sequences, novel peptides, and it really doesn't allow us to correlate what we may expect to what we see in the mass spec data. So basically what Peaks is trying to do then to look kind of beyond that traditional proteomics into what some of our customers refer to as a dark proteomics. Now we also know that the human genome encodes thousands of non-canonical open reading frames in addition to those protein coding genes. And in fact, many of these ORFs are likely found in immunopeptidomics samples. And we suspect that many of these could be responsible for mediating cancer, disease responses, and really identifying these non-canonical HLA peptides could be the key to some of the at least medicinal and treatment developments. Now, using traditional database search tools, identifying these is not possible for a couple of reasons. For example, even if you use a translated non-canonical database as concatenated with canonical, your immense search space will likely kill your FDR control and your result will be poor, of low confidence. And another thing is that the novel peptides, which are not present in canonical or non-canonical database or mutated peptides, will be completely omitted. So how can Peaks help to kind of bring that immunopeptidome discovery to another level and try to better integrate the canonical, the non-canonical, and novel proteome or a peptidome? So we have developed a really unique but also very complex workflow to try to help the immunopeptidome community. And in this workflow, so in the background, we kind of do multi-round searches. We start off with a DeepNovo, so we know we have our great GraphNovo sequencing algorithm, and we sequence all of the MS2 spectra. And once the sequencing is done, we move on to the first round search against the canonical database first. Then we use our spider to do some homology search against the canonical database. Then the second round on unmatched peptides against non-canonical, and spider against non-canonical database. And essentially in the background, we actually combine all of this. We create an internal FASTA, and we then research everything in the same fashion. So we do another really domain search right here, and then we apply the FDR control to finally report canonical peptides, non-canonical peptides, homology peptides, again, either against non-canonical or canonical database. And again, because we do have our sequencing algorithm, we do report these novel peptides, which of course would have some sort of high score. And if you wish, you can do some LFQ and of course some QC on your data set. In the workflow, what this looks like, essentially, it's kind of familiar, except that in the database selection, you can now define the target database, which would be, let's say, some sort of canonical database, non-canonical database, in addition to a contamination database. And this second database could be really any other uh, any other database that you wish to search in addition to the target and in the second round fashion. And Satya is here. I know he's using the bacterial database, which again, this is where you can try to expand that search space. Of course, for the peptide length, it's particularly useful to have it here to be able to define this for HLA peptides. The one last thing that I'd like to point out, I don't know how many people have used it. Yes, this was added in our latest version. It is a personalized on-the-fly training. And basically, once we have the identification result, we reevaluate this result based on the training on your given data. So we look for motifs within your samples or experiment, and then we recheck if there were any candidates where the scoring could be altered and a more personalized result produced. And finally, to kind of bridge that proteomics and genomics, we introduced a gene tab in our results, and it really is intended to kind of give user an easier visualization option or easy validation of these HLA peptides originating from either canonical or non-canonical regions. If you have a gene site information within your FASTA, we'll create this nice little chromosome here. Will indicate, I don't know how much you can see in blue here is the actual gene on the chromosome, but really again what matters is this the overlapped regions from the canonical and non-canonical proteins, where we can now nicely point out the peptides that we could actually identify, and then DeepNovo will also be listed under the protein gene if it has a certain tag sharing. So we know that these are just the DeepNovo peptides, they're just the sequence peptides, but if they have a certain, and by default it's 5 or more amino acid sequence sharing with the protein, then we'll kind of list it under this gene tab in the same kind of visualization here. And then, of course, we give you the statistics in terms of, again, database search, FDR controls, canonical peptides, non-canonical peptides, homology peptides, and DeepNovo peptides. If you click on any of these, really, we will give you the actual spectrum. Either you can flip here between the mirror plot or the actual assigned pre-processed MS2, that is just the empirical MS2. So with this, I kind of completed my story on the current peptide sequencing algorithms and how we integrated these for increased confidence and proteome coverage for both traditional and non-traditional applications. And I'd just like to quickly give you an insight on some new developments and what you may be expecting in the future from us, and then finish off with some final notes. So here's a very new January 6th dated publication by Dr. Ming Li, who is the lead researcher and actually the president of our company, if you did not know, and the owner of BSI. So with his, I believe, postdoc, they published and described a new approach to classical database searches now with deep learning fully incorporated. So I guess the idea is to one day replace the traditional de novo assisted classical proteomic searches. And essentially, some of the key features are listed here. They call it a deep search engine. This is really above my head, so not to go in any really details, I'd like to just essentially tell you that they developed an approach that they claim to have an improved scoring with less bias, especially for PTM identifications, and with a higher proteome coverage. And I just want to show you this one figure from the paper where they really looked at the score basically as calculated by the search engine and then versus the expected and the calculated PP. You can see the consistency of, the deep search engine across these scores, a really nice number of PSMs compared to some other search engines, and also across, again, different samples. We are, again, talking about deep learning or AI fully incorporated here. So I'm sure they're going to keep working on this approach, and hopefully it will be implemented in some sort of near future in our Peaks software. And just to finish off with some final notes, the excuse me, the peptide sequencing algorithms that I showed you, both as standalone and as integrated into different workflows, were based on the DDA data. However, we do have DIA sequencing algorithms developed as well. So we have our DIA DeNovo for some general proteomics, which typically DeNovo step here follows the identification step. So either library or database or combined library and database searches. Then we do DeNovo sequencing on the leftover unmatched precursors here. DIA is also implemented for peptidome searches with, again, DeepNovo assisted. And in addition to our Studio or Peaks Online, of course, for PeaksAB, our sequencing software, DeNovo is absolute key to sequence multi-digest samples to align those sequences and derive finally the protein or antibody sequence. And the very last point is our glycan finder also uses uses deep learning for sequencing, but not of the peptide part, but of the glycan part of the glycopeptide. So glycan finder can determine glycan compositions and structures using our deep learning algorithm to report novel glycans not found in the glycan database that was used for the search. And really the reason why I'm mentioning this is that in 2 weeks, we will have a webinar talk about the glycan finder. And specifically, I'm really happy to say we're going to have a very special guest from SciEx who will be talking about their EAG fragmentation for increased sensitivity for glycopeptides identifications and characterizations using Peaks Glycan Finder. So join us if you can. We will have only one session just because of the time zone that he's in. But with this, again, if you have any questions at any time, feel free to reach us out either directly or through our technical support email. Sales, if you have those questions, or of course, if you have any need for lab services, feel free to reach us out. And with this, I'd like to thank you again for your time in this kind of late afternoon. Thank you again, and I'm happy to take any questions if you have.